I'm really delighted to welcome and introduce Jim Shorter today. Uh, Jim is a professor of biochemistry and biophysics at University at UPenn. Jim originally he's from the UK. He did his grad school, his undergrad and grad school there at Oxford and University College London, and then he subsequently moved to the States, where he spent some time at Yale and then at MIT working with Professor Sue Lindquist, and was really one of the first people that utilized yeast to as a model to look at neurodegenerative diseases. Jim is a leader in this fast progressing field of phase transition. He's made seminal contributions in the context of how neurodegenerative associated proteins like FUS and TDP-43 play a role in the formation of these membranous organelles. He's also made great contributions at new innovative technologies to reverse some of these transitions. And I'm delighted he's here, and I'm looking forward to his talk. So, Jim, welcome.
Great. So thanks so much to the organizers for the opportunity to present to you today. And I'm excited to tell you about some of our latest thoughts on how we might counter some of these deleterious phase transitions in ALS and FTD. And I'll explain exactly what I mean by that. So the target I'm going to focus on today is TDP-43, which we've heard a lot about already. This is an RNA binding protein with a prion-like domain that typically we find in the nucleus, but in the context of degenerating neurons of ALS patients, we find that TDP-43 has become depleted from the nucleus and has accumulated in these cytoplasmic inclusions. Now, this is a very common phenotype in ALS. About 97% of ALS cases present with this type of TDP-43 proteinopathy. And my simple-minded view of this disease is that we'd really like to get TDP-43 out of these inclusions and put it back in the nucleus. And if we could do that, I'd anticipate that this would be something highly therapeutic for the disease. TDP-43 proteinopathy doesn't only happen in ALS, we also see it in about 45% of FTD cases. So let me tell you a little bit more about TDP-43. So this is an RNA binding protein with a prion-like domain. And typically it resides in the nucleus, but it does shuttle in and out of the nucleus. So there's always a small cytoplasmic pool of the protein. It engages RNAs through two RNA recognition motifs and really plays an important role in a variety of functions in the nucleus, including pre-mRNA splicing, transcription, RNA processing, and RNA transport. And is really responsible for regulating the expression of thousands of human genes. So there's a definite thought that the loss of TDP-43 from the nucleus, this loss of nuclear function, is contributing to the neurodegeneration we see in disease. So in degenerating neurons, we find it in these cytoplasmic aggregated structures, and mutations in TDP-43 are now well-established causes of ALS and FTD. Now, interestingly, almost all of the disease-associated mutations cluster in this region of the protein that we call a prion-like domain, which is a very unusual low complexity domain that is enriched for only a few amino acids. So we typically only find uncharged polar amino acids like glutamine, asparagine, tyrosine, and serine in this domain. And then aside from that, the only other amino acid that we see very much of is glycine. And so this type of amino acid composition really reminds us of domains that we know about in various proteins in yeast that are able to form prions, so infectious proteins. And having the presence of this type of domain is all you need to be able to form a prion in yeast. And we're very curious if the same applies to TDP-43. So let me tell you a little bit more about the prion-like domain. So almost all the disease mutations are found in this domain. But over the last 10 years or so, I guess, what we've appreciated is that the prion-like domain of TDP-43 also enables the protein to undergo this process of liquid-liquid phase separation, where TDP-43 is initially found in a monodispersed state in solution, but through transient contacts between prion-like domains and other domains of the protein, can condense into a liquid form of TDP-43 that we think underpins the formation of various functional membranous organelles inside the nucleus. And so this, it turns out, is a very important aspect of TDP-43 biology. TDP-43 has to undergo this process of phase separation in order to conduct some of its RNA processing reactions. And if you take away the ability of TDP-43 to form these liquid droplets, you lose expression of various TDP-43 target genes. So this is a functional aspect of TDP-43 biology, but at the same time places TDP-43 at risk from forming more stable structures. And over time, these liquid forms of TDP-43 can transform into more gel-like structures, and ultimately the type of cross-beta structures, amyloid-like structures that we associate with the end stages of disease. And it's this step of this phase separation landscape that we term an aberrant or deleterious phase transition. And it's typically the step of this process that's accelerated by the disease-linked mutations in the prion-like domain of TDP-43. So one question we had is this aberrant phase transition of TDP-43 really neurotoxic? And so to get at that question without having to massively overexpress TDP-43, we collaborated with Chris Donnelly's lab at the University of Pittsburgh. And Chris had taken a very clever approach to tag TDP-43 with this Cry2 protein tag. That is a protein that originates from plants and clusters in response to blue light. So if you shine blue light on this protein, it will oligomerize. And if you take the blue light away, it will then disperse. But in the context of TDP-43, if we put this tag on TDP-43 in response to blue light, TDP-43 will cluster. But that when we take the blue light away, TDP-43 will now have formed these more stable structures that don't spontaneously disperse. And we believe we can actually induce TDP-43 to form disease-relevant structures using this technology. And so if we do that in cortical neurons, here expressing opto-TDP-43, as we call it, in the absence of blue light, what we see is TDP-43 is forming these phase-separated condensates inside the nucleus, as we expect it should. These are functional forms of TDP-43. But if we shine blue light on these neurons, what we see is that after a few hours, we begin to notice that TDP-43 has accumulated in these cytoplasmic structures that initially have liquid-like properties, but over time transform into gel and solid-like states that lose dynamics. And it's about that time that we see this aberrant transition from liquid to solid states that the neuron begins to get into trouble. And this neuron is dying off at these later time points. Remarkably, these cytoplasmic inclusions of TDP-43 bear striking resemblances to properties that we see in disease-associated aggregates. So we see various PTMs, also recruitment of endogenous TDP-43 to these structures that really mimics the type of TDP-43 proteinopathy that we see in patients in degenerating neurons. And moreover, it's around the time that we get this aberrant phase transition of TDP-43 that the neurons really begin to get into trouble and start to die in the culture. So we really think that this aberrant phase transition of TDP-43 is something that's very damaging to neurons. And so one thing that my lab is very interested in doing is finding ways to reverse these deleterious phase transitions as a possible therapeutic strategy for ALS and FTD. And so we have several modalities that are capable of doing this now in the lab. And today I want to focus on one specific modality, which we call bait RNAs, which are short RNA sequences of about 20 to 35 nucleotides that engage TDP-43 and have shown a remarkable ability to actually drive this phase separation landscape in reverse. So taking TDP-43 out of these stable aggregated structures, but also out of liquid forms, and back to monodispersed TDP-43 that we think we can then transport to the nucleus and restore nuclear TDP-43 that we think will be a therapeutic thing to do in the context of ALS. So this idea originated through several different pieces of evidence, but my lab's very much a pure protein biochemistry lab. And one of the things we noticed when we tried to purify TDP-43 from E. coli is that the protein is very aggregation prone, intrinsically aggregation prone protein. So typically we purify TDP-43 with a large solubility tag like GST or MVP to keep the protein soluble. But as soon as you cleave that tag off of the protein, TDP43 very rapidly aggregates in the test tube. And so we wondered whether we could antagonize this very rapid aggregation of TDP43 by simply including bulk RNA in this reaction. And so here, just including total yeast RNA in this reaction, we very potently stop this spontaneous TDP43 aggregation process. This RNA binding dependent effect required that the RNA to engage the RMs of TDP43. So if you mutate 5 phenylalanines that engage RNA in the RMs of TDP43, then TDP43 will now also aggregate even in the presence of RNA. So RNA has to come, engage the RMs of TDP43 to stop this aggregation process. So we can track this in another way. So if we just track the aggregation by turbidity, we see that RNA is very good at stopping TDP43 aggregation unless it bears these mutations in the RMs of TDP43, which don't engage RNA, and the protein is then resistant to the effects of RNA. We can set this up in a slightly different way. So if we include RNA in the reaction, allow TDP43 to incubate, we notice that initially there's no aggregation of TDP43, but if we add RNAs to then degrade the RNA in the reaction, then TDP43 will very rapidly aggregate in the test tube. And so we thought, hang on a minute, isn't this a therapeutic opportunity? So could we find short, specific RNAs that also elicit this type of effect? And what we'd really like to find are short RNAs at about 20 to 35 nucleotides in length that antagonize this aberrant TDP43 phase separation. And the reason we want RNAs of this size is that these are similar in size to an antisense oligo that can be readily delivered to the CNS of patients to exert therapeutic effects. So we're very keen to find RNAs in the size range that might also inhibit and antagonize TDP43 aggregation. And so if we could find these, could these also be developed ultimately into therapeutics for ALS and FTD? And so we've begun with this lead bait RNA that we call CLIP34. This is a natural TDP43 binding sequence that you find in the 3' UTR of TDP43's own mRNA. And it's this region of the TDP43 mRNA that TDP43 engages to repress its own translation. So if we take this isolated piece of RNA, 34 nucleotides, we actually find that it's a very potent inhibitor of TDP43 aggregation. Even at substoichiometric concentrations, we can completely eliminate TDP43 aggregation in the presence of this short RNA. The IC50 for the reaction is about 0.5 micromolar when we have 5 micromolar TDP43. So 10-fold excess of TDP43 over RNA can still be inhibited by this short specific RNA. Next, we wondered whether this also required engagement of the RRMs of TDP43. So this mutant form of TDP43, 5FL, was resistant to the effects of CLIP34. We could not inhibit the aggregation of 5FL with CLIP34. But we also wondered whether any RNA could also exert this effect on TDP43. And so this turned out not to be the case. If we take this control RNA, AC17, this is unable to antagonize TDP43 assembly, suggesting that we really are able to find short specific RNAs that are capable of inhibiting TDP43 aggregation. So next, in collaboration with Chris's lab, we wanted to know whether we could exert therapeutic effects in our optogenetic model of TDP43 proteinopathy. So in this experimental paradigm, what we're going to do is we're going to express the opto-TDP43, and then we're going to treat cells with the oligo and switch the blue light on to stimulate the aberrant phase separation of TDP43. And here we have a scrambled form of CLIP34 as a negative control. So if we do that experiment, shine blue light onto the neurons in culture, what we see is that with the scrambled control, we begin to see the cytoplasmic inclusions of TDP43. But this phenotype is completely abrogated in some cells, where we keep TDP43 in the nucleus and stop it accumulating in these cytoplasmic aggregated forms. And so that's just quantified here. We also have a very strong protective effect. So these neurons that keep TDP-43 inside the nucleus do not experience cell death as much as in the scrambled control. So this is a neuroprotective activity of this short RNA. But of course, you'll recognize that this is a prevention paradigm. And what we'd really like to do is target these TDP-43 inclusions once they've already formed. And so in this experiment, what we're doing is we're pre-assembling the recombinant TDP-43 into aggregated forms, and then simply just treating with CLIP34 or the AC17 control. And what we see is that we are actually able to partially dissolve these preformed inclusions of TDP-43 with CLIP34, but not with AC17. We're actually able to recover about 20 to 30% of soluble TDP-43 from these pre-aggregated structures. So that's in a test tube. What about in our optogenetic model? So here we're going to change the paradigm and shine blue light on the cells first to allow TDP-43 inclusions to accumulate in the cytoplasm, and then treat the cells with RNA and ask, can we resolve those aggregated forms of TDP-43? And so if we do that here with a control oligo, here's a cell that's experiencing TDP-43 proteinopathy. And after several hours in the culture, this cell rapidly dies off. And this is a typical observation with cells experiencing massive TDP-43 proteinopathy like this. By contrast, if we treat these cells with the CLIP34 RNA, here's a cell that has pronounced TDP-43 proteinopathy and even depletion of TDP-43 from the nucleus. But upon treatment with CLIP34, we're able to solubilize this cytoplasmic aggregated form of TDP-43 and put it back inside the nucleus. This turns out to be something that's highly protective to these cells in culture as well. And so, boy, would you like to be able to do that in ALS patients, right? If we could really get TDP-43 out of those aggregated structures in the cytoplasm and put it back in the nucleus, we'd anticipate that this would be something that would be highly therapeutic. A natural question that arises from these studies is, can we also restore the functionality of TDP-43 inside the nucleus so we can show that TDP-43 gets back to the nucleus? And in the experiments that are going on now, we're testing whether we're really recovering TDP-43 functionality in the nucleus, and those experiments are looking good, and I think we will be able to restore TDP-43 function. But this also raises the question, is this something safe to do? So what you wouldn't want is a situation where the bait RNA remains so tightly bound to TDP-43 that it never comes off and inhibits TDP-43 functionality inside the nucleus. So ideally, what we would find is a bait RNA that transiently engages TDP-43, takes it out of the aggregate. TDP-43 then is restored to the nucleus, but then the bait RNA no longer engages TDP-43, and TDP-43 can go and perform its regular function inside the nucleus. So we did a number of experiments to assess the safety of these bait RNAs. First of all, what is the localization of these bait RNAs when we introduce them to cells? And what we find is that we primarily find them in the cytoplasm of cells at early time points, suggesting that they wouldn't interfere with the nuclear function of TDP-43 because they're simply not in the right place to do so. Moreover, if we track the localization of TDP-43 treated with bait RNAs, we find that this doesn't affect TDP-43 nuclear localization. So we're not perturbing TDP-43 biology in that way either. Moreover, if we try and track TDP-43 functionality in cells treated with these bait RNAs, so here in the exon skipping activity in the CFTR gene, if we knock down TDP-43, then we get a pronounced phenotype in this assay, but the bait RNAs don't induce the same effect, suggesting that TDP-43 is maintaining functionality even in the presence of these bait RNAs, indicating that this could be something safe to do. So CLIP34 is our initial lead bait RNA, but we've been exploiting a lot of RNA-seq iClip data, looking for bait RNAs that work even more effectively than CLIP34. We actually have a number of these now that work more effectively than CLIP34 against TDP43. And they don't only work against wild type TDP43, they also work against a variety of disease-linked mutant forms of TDP43 and also various PTM mimetic forms of TDP43 that we associate with disease. So I'll close there and conclude. So under ordinary circumstances, we find TDP43 inside the nucleus, engaging RNA and performing its biological function. But in the context of degenerating neurons of ALS and FTD patients, we find that TDP43 is depleted from the nucleus and has accumulated in these cytoplasmic aggregated structures that are actually relatively devoid of RNA. So these aggregated forms of TDP43 don't contain much RNA bound to them. And so what we think we can do with the bait RNAs is solubilize these TDP43 inclusions by engaging the RRMs of TDP43. This liberates a TDP43 that's bound to the bait RNA. But then we believe an interesting aspect of TDP43 biology kicks in, where the nuclear import receptors come and engage the NLS sequence of TDP43 to transport it to the nucleus. And one of the functions of these proteins is to eject any bound RNA on TDP43 such that you take an APO form of TDP43 back to the nucleus. So in this way, the bait RNA would get recycled for further rounds of action upon the aggregated forms of TDP43. And we have a pseudo-catalytic protein disaggregate system encompassed by these bait RNAs and the nuclear import receptor system. So I've told you that bait RNAs can prevent TDP43 inclusions, reverse TDP43 inclusions, and protect human neurons in culture. We've also taken a similar approach with the FUS proteins, and we also have now potent bait RNAs that antagonize the assembly of FUS and ALS-linked forms of FUS. And the goal is now to optimize these bait RNAs and try to take them to animal models. So can we treat an animal model of TDP43 proteinopathy or FUS proteinopathy with this type of bait RNA technology? And so Chris and I have been excited to spin out a company called Confluence Therapeutics, where that is their immediate goal, to try and treat mouse models of TDP43 proteinopathy, for example, with this bait RNA technology. So I'll stop there. I'm very grateful to my lab, a number of great collaborators, including Chris Donnelly and Jake Mann, who's in the room here, and various funding sources. And I'm happy to take any questions.
That's amazing. Do you have to worry about an innate immune response to these RNAs, or is it seen as self?
Yeah, good question. So I think that is something to worry about. So we would envision perhaps including pseudouridine instead of uridine in the RNAs. But ASOs face the same problem, right? And somehow they evade the innate immune system as well. But it's a similar issue.
Is there a way to make them like drug inducible? So like it folds on itself, you add the drug, it opens up, it does its thing, and then remove the drug, it now gets into a quiet mode?
Yeah, that would be awesome, right? I guess we'd need AAV technology to try and do that sort of thing. Yep.
Hey, Jim, great work. So you showed us a lot of beautiful systems where these beta RNAs were able to reverse TDP43 functions. I'm wondering whether you've been able to assess their efficacy in physiological systems like iPSC models, where you have endogenous TDP43. And are you concerned at all? You kind of hinted about the potential toxicity of adding these molecules, particularly in the physiological condition where you don't have aggregation of TDP43 or dysfunction.
Yeah, so those are experiments we're conducting now. We have more advanced data on that area with FUS and various iPSC neuronal lines. And there, the lead FUS bait actually does quite well in mitigating some of the disease phenotypes in iPSC neurons. But yeah, I agree that's an important next step for this project.
Great. And then a slightly different question. Do you think it's the sequence or the three-dimensional structure of the bait that determines its specificity? Or I guess are both equally important in your experience?
Yeah, great question. So many of the baits that we have uncovered do have this distinctive stem loop structure to them. And one of the things we know TDP-43 does when it engages that structure is actually unfold it. So it relieves that stem loop structure. And we're exploring whether that's an important aspect of their activity.
Thank you.
So it's amazing work. It may be changed the world of ALS. My question is, how do you expect is the RNA, the 34 nucleotide RNA, is that stable in vivo? How do you think if you are going to test it in the animal, how long do you expect it's going to effective, and then how many doses do you expect to inject for the course?
Yeah, it's a great question. And this is an important question in the ASO field as well, is how do we make the RNA stable enough that they can exert a therapeutic effect in vivo, and it's a similar issue for these bait RNAs. So through various RNA modifications and things like that, we would look to enhance the stability of the RNA in vivo.
Yes, great work. I was thinking more about the prion disease, when there is a shift between the protein, the healthy protein with the alpha helix, towards the protein that is pathological, more with the beta helix. And I was wondering if this RNA will work like sticking to the protein, to disaggregate, and unfold whatever is going on. And if you think you can apply the same for other disease.
Yeah, I mean, I think that's exactly what we think the bait RNAs are doing. So they're engaging the RRMs, and somehow, through a mechanism we don't understand, that is eliciting an allosteric effect on the prion-like domain and altering the folding of the prion-like domain. We're excited about this for RNA binding proteins like TDP-43 and FUS. A question is whether we could extend this to other RNA binding proteins, like tau, for example, is a prominent RNA binding protein. Yeah, that's something we're interested to explore.
Okay, thank you.
So maybe I can ask a couple of questions that came through the chat. So how does this relate to selective neuronal vulnerability? And, within the context of selective neuronal vulnerability, how would you explain this phase transition? Does it happen in neurons that degenerate or globally? And knowing that TDP pathology is observed in many diseases, would you comment on that?
Yeah, I guess this ties into TDP-43 loss of function and how different neurons may have differential sensitivity to TDP-43 loss of function. That would be my sort of initial thinking on that. But then why does a patient get FTD rather than ALS? Is it just the neurons in which it starts in first, for example? These are interesting questions. And
the other one, what is the role of autophagy and mitochondrial dysfunction within that picture? Do those happen before and then you have phase separation, or you have phase separation and then these occur?
Yeah, great question. My sense would be that the aberrant phase transitions are contributing to the defects in autophagy and also mitochondria as well. But that's just my bias. I don't really have any evidence for that.
So then I asked the question, what experiment do you think we should do to find out the answer to that question?
Yeah, I mean, I think a time course of exactly what is changing, for example, in our opto TDP-43 models, that would be interesting.
Yeah,